how valuable is the associated clinical information? Is it more valuable than what we've just said in the long QT? Have you had your chocolate cake and wine?
Dangerous. You're right. I think it is different. Not that clinical information is not helpful. Of course, it is, including those interpreting the genetic tests, because the variants of unknown significance are just as important in hypertrophic cardiomyopathy than they are in any of these other diseases. But the problem is that knowing the wall thickness, for example, in this disease is not probably going to be helpful in deciding whether that's a disease-causing mutation or a benign polymorphism. It's a very diverse disease. There is no wall thickness that's not compatible with hypertrophic cardiomyopathy. And I think that it probably is a different situation than, for example, LQTS. Do you agree with that?
Well,
yeah.
You're the expert on
yeah. Thank you.
Just to complete the cycle, maybe Charlie Antzelevitch would want to comment on his thing. And I hope we're not pulling people away, the tables away from discussion, but this came up and seemed to be a terribly important part. So, Charlie, if you haven't had too much wine and chocolate cake, maybe you could make a comment on the areas that you're actively investigating now.
I'm known for my brevity. Ditto. Well, I think the important points have been stressed already, and that is that when it comes to the variants of unknown significance, it's the co-segregation of the gene with the phenotype that's very important, and that can lend more significance to it. But the only thing that I would add is that we have a few people in this room that do expression work. And it's important at this point to take that variant and to do heterologous expression to see whether that genetic variation or mutation does produce a loss of function or a gain of function that is consistent with the phenotype, and that lends additional significance to that particular variant. That coupled together with the co-segregation of the gene and with other data that will become available as the years progress is going to take us from the VU to the definitive diagnosis. I think Arthur has more to say.
I just thought that we have such a group of experts here that can answer some of the questions. Craig, do you want to make comment at all on the expression studies, or why don't we have the Wisconsin group come up here and let them, and then we'll get back to eating. Could we get the two Wisconsin, Craig, you have to come up too. Huh? Okay. It's here.
Arthur, thank you. John Mikulski and I did come for the wine and the dessert and not to talk. But I will just echo a couple of points. One is a clinical cardiologist, because we see patients with these diseases, mainly ion channel diseases, and I sometimes say that as we genotype patients, it's the polymorphisms and the variants of unknown significance that are the big problems. They're the ones where telling patients what they have becomes a problem. And so it's interesting to me to see this as an area that continues to expand. And secondly, I agree with you that most gene mutations have never been expressed. And so when you think of this, the genotypers are much faster than the expression people. And so most mutations are not known from a functional expression perspective. We don't know what's wrong with these channels. We don't know what they look like. And secondly, they're expressed in heterologous cells, cells from kidneys and et cetera, but not from the heart. And so that may also be important. So we have a great deal to learn, and we're entering the stem cell era where we can begin to recreate heart cells from people that carry their genetic abnormalities, and there's almost no information about what this kind of science is going to teach us. So it's a wonderful field. It's clinically important. It's basic science important, and we've got a lot to learn. So anyway, Art, may I go back and have... Thank you.